super excited to get to kick things off. We're going to talk drug discovery with AI and ML. And as Martina introduced, I'm going to be talking about mapping and navigating biology and chemistry with genome-scale imaging. Now, using the map analogy and taking it maybe a little bit further than it should go, I'm going to draw the map of the landscape of what we're working on here at Recursion and give a really high-level overview and try to make sure that it's understandable and translatable. And then Federico and Oren are going to do some deeper dives into the really cool details of what they're working on. So hope you stick with it and interrupt, ask questions if you have any questions for me. All right. So I want you to all step back for a minute and imagine if one tool could show you the whole landscape of routes to the same destination, in this case, from the airport here to Recursion. And that same tool could show you ways to get around traffics and roadblocks in the city and give you different options for how to get there. Now, obviously, I'm just showing you things from Google Maps, which is a tool that we all have on these amazing pocket computers we carry around with us and that we really take for granted. But I want to pose the question of what would happen if we could do this for biology too? So just like a city, cells are these complex adaptive systems. If we perturb one part of the system, it causes cascading changes throughout the cell. In a city, if we have construction on one street, it causes cascading changes throughout that city and you get weird traffic patterns. If we perturb a cell by making it sick or with a gene mutation, then it causes all sorts of downstream changes as well. So how can we map this complexity and understand better what's going to happen if we perturb a cell in a particular way? In city maps, we talk about the distance between places and the routes that we navigate to get from one place to another. For biological maps, we have to think as well, what are the properties that we care about? And so in these maps, we talk about similarity. We want to know how similar or dissimilar two different biological states are. And intuitively, we think about this, if we make two different perturbations to a cell, if they make the cell do the same thing, then they should be similar. If they make the cell do opposite things, or if one reverses the effect of the other, then they should be opposite to one another. I'm going to show you an example of this mTOR pathway, which is a pathway that's really well studied in oncology. And I want to highlight a couple of things. One is that this tumor suppressor complex, which is made up of two proteins, tuberin and hamartin, it inhibits mTOR. And secondly, that if these two proteins are acting together, so we expect them to do very much the same thing. So if we then do a genome screen and we knock these proteins out, each one at a time, and we ask what happens to the cell, we can make a map, and this is what we would expect to see. We would expect to see that TSE2 and TSE1 are very, very similar to one another. If you knock one out, you break the complex. If you knock the other one out, you also break the complex. They should do the same thing. We should also see that they do the opposite thing of mTOR because they are inhibiting mTOR. And so if we knock them out, mTOR should turn on. If we knock mTOR out, it turns off, obviously, because it's not there. So they should be opposites. And this is a snapshot from our map, and it is actually the kinds of relationships that we can see and capture in these maps. So how do we start to understand what the state of a cell is? So ideally, we would have one assay. We would have one assay that we could use for any perturbation, regardless of what it is, and we wouldn't have to design one for each gene or for each chemical we care about. An assay like this would be useful if it covered a whole breadth of perturbations. It would be useful if it was standardized, so we didn't have to change it very much from one perturbation to another. And it would be useful if we could compute on it, if we can easily analyze and integrate that data from every perturbation. Now, molecular omics come pretty close to this. They're great high-dimensional spaces. They're easy to interpret, but they are really expensive. It's really hard to do this across millions of compounds and thousands of genes. But imaging, okay, after a brief interruption, let's come back to why imaging is cool. All right, so imaging is a downstream readout. If we perturb the cell, if we knock something out, if we throw a chemical in there, what we see in the images is downstream of all the changes that have happened. So it gives us a good sense of like, what is the complex effect that this perturbation is causing in the cell. It's also really data rich. We get a lot of information from a single image, and it's cheap. They're way cheaper than running many of the omics technologies. And so what we do here at Recursion is we use a standardized assay that we call phenomics. So 6 different common cellular substructures get stained, each with a different dye. And then we put those together and look at those images. And what we see is we can start with a particular disease model where we've perturbed these cells to make them sick in some place. We can compare them to a healthy control. And we can also absorb differences when we treat with different compounds. So that tells us we're seeing a readout that's telling us meaningful things about biology that's happening. So with this capability, we were able to ask this question. What happens if we take the cells and we knock out all the genes in the genome, or we dose these cells with hundreds of thousands of molecules at lots of different concentration? Can we build a map of biology from these pictures? So the first challenge that you should probably all be thinking about is that it's really hard to compute on image data. Image data is just a set of pixels. And so we don't know what's important for biology or what it means for one image to be opposite to another. The similarity is reasonable, but what does it mean to be opposite to an image? We started out by computing on these images with feature engineering. And I think that Oren will talk through a lot more of this evolution in detail, but we started with these human engineered features. And then we shifted to using deep learning models. And the reason is that these models are able to turn unstructured images into computable data in a way that's a lot faster and more reliable than human features. So what we do is we train these multi-class classifiers on these images to identify what we did to a cell. We take a picture, we say, what did we do to this cell? And we learn features from that. And those features are actually biologically meaningful. And these models can help us to correct for things like batch effect. So the one challenge here is that these features are typically uninterpretable, but that's totally okay, because what we're doing is we're using these features as a space in which to compute similarity or dissimilarity of images. And that is what we use to make the maps. So we don't need interpretability here. More recently, you may have seen on Twitter some of our posts that we've been moving from these multi-class classifiers to foundation models. And I am absolutely not going to steal Oren's thunder, other than saying what we're doing is we're taking this crazy thing of taking an original image, masking out 75% of it, and then saying to the model, tell me what that original image looked like. So these maps of biology and chemistry are a great tool, and they're sort of core of what Recursion does. But the question is, do they work? And what can we do with them once we've made them? So we've tested these maps of biology in a variety of different contexts, and I'm going to talk through them rather quickly. One of the applications that we've used, if we started with a query in our map of what are the genes associated with breast cancer? And with no other prior data, our map is able to recapitulate these relationships between genes involved in breast cancer and reconstruct decades of known relationships. We've also used this to drive programs. And so recently we released some information about our Target Gamma program. This program started with this CDK12, which has been advanced as an idea for improving response in this HR proficient setting. Now, the problem is that CDK12 is very similar to other CDKs, and so getting selectivity is really, really challenging. We were able to use our maps to identify a target, RBM39 here, which is very similar to CDK12, but which isn't very similar to CDK13. And we can find these compounds that target and are very similar to both of these proteins, but that don't have that same similarity or that same impact on CDK13. And so that lets us work around things that were previously thought to be undruggable because they just weren't selective enough. The final thing, which I think is probably the coolest thing in my perspective, is that this screening means that we don't get the similarity of a compound to just one target. we get the similarity of that compound to all targets. And that means we can do really interesting things and ask really interesting questions about polypharmacology and toxicity. In these plots, what we see is a compound bortezomib, and it shows similarity to the protein or to the proteasome subunit knockouts. And it also shows similarity to the splicing factor that's known to regulate those things. And so we can monitor as we're trying to optimize compounds, what's the activity across all of these targets instead of being limited to lower dimensional assays. So with this, there's this question of what does drug discovery look like in the future? We've shifted a lot from talking about being a biotech company to being a tech bio company. And the reason is that we really want to bring some of the ways of thinking that the tech industry has into the biosciences. One of the ways that we do that is by partnering. We partner both with people in the AI space and in the tech space and with those in pharma. So right now we have 2 partnerships with big names in pharma, and I'm not going to go into the details here, but I think the big advantage that we get from these partnerships is that we understand how pharma thinks, we understand what the key questions are that we need to be addressing, and we understand how to communicate and validate what we're doing. We've also recently talked about this NVIDIA partnership, which gives us the ability to do a lot of compute on their platforms and lets us drive these foundation models for biology and chemistry that you'll hear more about later tonight. And really the reason is that this is the bitter lesson about AI research, which is that if you want to get good AI models, you have to have both big data and big compute to compute on that data. And so this is from a paper or from an archive paper that we put forward for a recent conference that shows that if you use public data and small models, you get the lower left corner there. If you use our private data set on small models, you get moderate recall as well. But you get the big gains in recall when you start upping the level of models. And so for this reason, we need to bring together both the pharma perspectives to know what it is that we're trying to find, and the AI perspectives and compute resources in order to build these big models. So the other thing that we're bringing to bio from tech is this commitment to open science initiatives. We've seen across a lot of AI fields that the way that a field moves forward is through good benchmarks, open data sets, and strong communities in academia and in industry. And Jonathan did a great job talking about Valence Labs and some of their commitments. I want to highlight as well our RxRx3 data set, which I think is really cool. It is 2.2 million samples of the data that we've generated. It's the largest, as far as we know, publicly released data set of perturbative cellular imaging. And we hope this is a way to start driving advances in machine learning technology, not only within Recursion, but in the outside community as well. This is also a fun slide for me to talk about because when I'm in these talks and I see people doing all these cool things with data, I always want to play with that data. And so this is me telling you, here's the data to play with. Now, on the other side, if you're more on the bio side than the tech side, we also released this tool, MolRec, which shows off how we relate small molecule activities to each other and gives you the opportunity to play with sort of this pre-crunched map data to start to see biological relationships, biological insights, the way that we can explore in-house. And that's also open sourced. I won't spend much time on this because Jonathan talked about this a lot, but Valence Lab is our semi-autonomous research institution that is really dedicated to building this community, pushing the frontiers of machine learning, and helping us really shape a new industry. So all of you out there are probably asking, great, this is really cool work. What does this mean for me? I think there's 3 ways that that you can take away something from this talk. The one is the data that I talked about. There are 22 million images that you can take, play with, experiment with, build models around, and see how this actually works. The second way is the tools that I talked about. RxRx.ai MolRec is the platform that will let you explore these relationships in the same way that we do. And finally, Valence Portal is a way to build that community and really get involved with these other people in the room who are also thinking about these hard problems. I want to finish up by talking about a few of the interesting things we're working on and that we would love to have thought partnership in. In the data science and machine learning bucket, we're working on foundation models. Oren will talk about that. We're also working with how we bring large language models into this process of drug discovery. We're working on bridging digital chemistry and biology, and Federico will talk a lot more about that. On the software and data engineering side, I think we have a lot of really interesting problems. We have massive amounts of data, and so we need to figure out how to store and work with quadrillions of data points and petabytes of data efficiently. To do this in an industrialized, scalable way, we have to build reliable pipelines, platforms, and learning cycles. We're thinking about how do we orchestrate these pipelines that bring together the ML models on the computers, actual physical people's lab operations, and getting those compounds and those reagents in-house so that we're ready to run those experiments. And finally, in this miscellaneous bucket, we're also thinking a lot about how we lean into this open source side of things, build a bio-ML community, how we manage complex operations, supply chain, and planning at scale, and how we build financial models for how we do drug discovery in this new imagined world. So to wrap up, the key takeaway here is that by using AI and ML, we're able to take this image data that is unstructured, turn it into biologically meaningful representations, and use those to map biology. The vision that we have is to use these maps to really decode biology so that we can predict from any perturbation what's going to happen in the cell and how do we impact or reverse those sorts of perturbations. This is just the beginning, and I'm excited to see what we as a community largely can do. If you have any questions, this is my email. There's also an email for questions on RxRx3 and MolRic. And with that, I'm happy to address a couple of questions and then turn the time over to Oren and Federico to talk in more detail. Yeah.
On the data science side, are y'all working with a particular library?
You know, we have over the years developed a lot of in-house stuff. We also, so we pretty much a Python shop, right? We work in Python across the board. And we have a variety of tools that we use both from the open source side of things and our in-house side of things. Yeah. So for folks online, the question is, in the mapping that we're doing, at what level are we specifying the nature of our compounds? So right now in the mapping that we do, the compounds that we specify are, so we specify them by name or by ID, right? So there's a bunch of things that we have in-house in our libraries that are an ID. There are a bunch of known compounds that are a name. We are thinking about how to represent those compounds in a meaningful way through some of these foundation models. And there was a recent paper that we put out about MolE, which is a way of doing that. And I think that's the next step forward. Like, we would love to not have to run these experiments on all the compounds, but be able to sort of, from the structure, think about what are the relationships going to be? What do we predict is going to happen? Any other questions?